Okay, so get this. A new paper just dropped, literally a few days ago, that might completely change how we think about controlling inflammation. We're talking about a real molecular detective story that takes place inside our own bodies, and it has a twist ending you won't see coming. So let's dive in and uncover the clues. First, let's start with a simple idea. You can think of inflammation as your body's personal team of firefighters, right? When you get an injury or an infection, this crew rushes in to fight the problem, clear out all the damage, and help your body heal. It's an absolutely crucial protective force, one of our best allies, really. But here's the thing, and this is the huge paradox of chronic inflammation. What happens when that emergency response just doesn't switch off? The firefighters keep going, and their life-saving work actually starts to cause serious long-term damage. This is what leads to some of the biggest diseases we face today. And that's the big unsolved mystery scientists have been wrestling with for years. How does the body tell these inflammatory cells to stand down? I mean, what are the exact signals that tell them, all right, job's done, time to go home? Finding those signals would be a total game changer. So one of the key suspects in this whole mystery is a group of molecules called epoxy-oxylipins. Think of them as these tiny but super powerful messengers made from the healthy fats in our diet. We've known for a while that they help regulate the immune system, but exactly how they do their job in humans, well, that's been pretty murky. So it all really boils down to this one question. What specific signals are in charge of our immune system's cleanup crew? We're talking about a type of immune cell called a monocyte, which circulates in our blood and rushes to any site of injury. If we could just figure out that signal, we might be able to control the whole response. To crack this case, scientists came up with this really brilliant experiment. And they didn't do it in a petri dish, not in a mouse. No, they did it directly in human volunteers. This gave them a real-time window to watch inflammation actually happen. Okay, the plan here was super elegant. First up, they triggered a really small, totally harmless inflammatory response right on the skin. Then they gave some of the volunteers a drug specifically designed to block one particular enzyme. All the while, they were watching for those classic signs, redness, swelling, and taking samples to see what was going on at the molecular level. The specific enzyme they went after is called soluble epoxide hydrolase, or SEH for short. Now, what does SEH do? Well, its whole job is to act like a deactivation switch. It breaks down those helpful epoxy-olipin messengers. So the drug was designed to block this off switch, which in theory should keep more of those good guys signaling molecules hanging around for longer. All right, so the team runs the experiment, they collect all the data, and then, well, what they found was not at all what they were looking for. The first results were a complete and total surprise. You see, the thinking was, if you boost these good guy anti-inflammatory molecules, you should see less of the classic signs, right? Less redness, less swelling. But nope, that's not what happened. The redness, the heat, the swelling, it was all pretty much the same, whether you got the drug or not. It really looked like a dead end. But it turns out, the real story wasn't happening on the skin at all. It was hidden deep in the blood samples. While the drug didn't seem to do much on the outside, it had a massive, and I mean massive, effect on the immune cells circulating inside. Just look at this data. The number of a specific kind of monocyte, the intermediate monocyte, absolutely plummeted in the group that got the drug. And that was the big break in the case. The real magic of this drug wasn't about reducing swelling. It was that it completely stopped this one specific type of immune cell from even showing up. Suddenly, our molecular detectives had their prime suspect. Okay, so now they had their clue, a huge clue, but they still had to connect the dots. They had to follow those molecular breadcrumbs to figure out how the drug was actually connected to those disappearing cells.
And their analysis pointed to one specific hero molecule. It turns out, when they blocked that SCH enzyme, the levels of this one particular epoxy-oxylipin, it's called 1213 EpoM, just shot through the roof. So the drug wasn't just blocking an enzyme, it was amplifying this one specific messenger.
So the next question is obvious, right? What was this hero molecule actually doing? Well, they figured it out. It was targeting another molecule inside the immune cells called p38 MAPK. Now, the easiest way to think about p38 MAPK is that it's the go signal. It's the order that tells a basic classical monocyte to grow up and transform into that more inflammatory intermediate type. So when you put it all together, you get this amazing chain of command. Step 1, the drug boosts the hero molecule, 1213 EpiOMe. Step 2, this hero molecule then swoops in and blocks the go signal, P38 MAPK. And step 3, with that signal blocked, the basic monocytes never get the order to transform. Case closed. They had found the exact off switch for producing these specific inflammatory cells. And this is where it gets really exciting, because this isn't just a cool molecular mechanism. This has some huge, real-world implications for how we might be able to treat human diseases. You're probably thinking, okay, so what? They found a way to control one tiny little subtype of immune cell? Why is that such a game changer? Well, it's a huge deal because this specific cell, the intermediate monocyte, turns out to be a key bad guy, a known villain in a whole rogues gallery of chronic inflammatory diseases. When you have too many of them, they're linked to causing serious damage in everything from rheumatoid arthritis and lupus to HIV, tuberculosis, and even cardiovascular disease. They're the firefighters that are known to go rogue. And that right there brings our whole detective story full circle. By solving the mystery of how to control this very specific part of the immune cleanup crew, these scientists may have stumbled upon a brand new way to stop those firefighters from burning down the house in a whole bunch of different diseases. So, what started as an experiment that looked like a failure led to this incredible serendipitous discovery. And it leaves us with this one big provocative question. Could manipulating a single molecule that controls a single type of cell be our next great weapon in the fight against chronic disease? You know, it's a pretty exciting possibility to think about.